Hello, Professor and fellow classmates. This is Group 1 presenting our poster, Multigene Barcoding, Can the Botanical Genome be as informative as the human genome? One of the first cases that used forensic botany as evidence was the kidnapping of the Lindbergh baby back in 1935. In this case, the wood from the kidnapper's ladder was presented as forensic evidence, and that was largely responsible for the conviction of Bruno Hauptmann in the kidnapping and murder of the baby. Forensic botany remains an underutilized discipline of forensic science to this day, even though it can provide copious amounts of supporting evidence and insight into criminal investigations. The hesitancy to use forensic botany is largely due to the lack of successful identification of botanical evidence through traditional morphological characteristics. There's a technique called multigene barcoding. It is a novel technique that uses genetics, genomics, and bioinformatics of short DNA sequences to identify species. This technique can aid in solidifying and strongly supporting the identification and differentiation of fragmented and degraded samples of botanical species. The plant DNA barcoding is unaffected by external factors such as age and climate, which makes it reliable and a sensitive technique. However, it is a simple, reliable, and accessible method of identification that yields a higher percentage of species and subspecies identification. The protocols required for barcoding can process thousands of sequences and samples in a short time, which makes it an ideal technique for identifying crime scenes and dump sites. The future development of the technique and the creation of a national database will aid investigators in identifying degraded and fragmented samples of botanical evidence at a faster rate. When using the multigene barcoding technique, we can develop a CODIS-like database for plant DNA called CBDIS, or Combined Botanical DNA Indexing System. In order to test our theory, we'll be using the Kaigen DNeasy Plant Mini Kit to test the single band formation in a 2% agarose gel, and then push through the BLAST algorithm for a species match. The results will then be used to compile our botanical identification database, or CBDIS, which will prove that the botanical genome can be as informative as the human genome.